It is my pleasure to introduce Katrina Rogers.
Thank you, Professor Wilkinson. I really appreciate the opportunity to speak to the class today. As he mentioned, my name is Katrina Rogers, and just as a lead-in, I'm a quality and leadership expert with over 25 years of experience in pharmaceutical and medical device discovery, development, and manufacturing. That sounds like a big thing, but I followed a winding path to get there, and it started here in Spokane. So first, I first want to share with you that I recognize that I'm a child of privilege. My family was not wealthy, but my parents always pointed me towards college. And I think that in today's world, if we don't acknowledge where we came from and what benefits we accrued from that upbringing, then we're not doing a very good job of starting on our path towards making a more just and civil society. So given that, and given that my family my father was in the military, he was an armed forces officer in the Air Force. So we traveled quite a bit, but family was always here in Eastern Washington. My grandparents lived in Newport, as a matter of fact. And so when he retired, we moved back to the Spokane area, and I went to high school at Riverside High School, which many of you may be familiar with, just a little bit north of where we're at here in Whitworth. I graduated when I graduated from high school, I did not immediately go to college. Instead, I chose to marry my high school sweetheart. And we spent a few years after that doing a number of things, some of which included post-secondary education. We both went to Spokane Falls Community College. And he went on to Gonzaga with an Army ROTC scholarship. And I followed him, of course, as an Army officer's wife, which really was my first calling. And I must say that I enjoyed it and I did it very well. But we made a choice after his 4 years of tour was up that my education was just as important as his. And so we returned to Spokane and then subsequently to Seattle so that I could attend school at the University of Washington. I've always been interested in chemistry. Ever since I was a little girl. My mom was a medical technologist and worked extensively in the chemistry unit at Sacred Heart Hospital here in Spokane. And so I majored in science at Spokane Falls Community College and went on to a chemistry degree at the University of Washington. My grades and my work at the Falls gave me the opportunity to have a scholarship at the University of Washington, which was of great help because that allowed us to be students in the campus area without having a tremendous amount of difficulty. Nevertheless, I still had to work full time while I finished my degree. And that was not trivial. I needed to work at least 35 hours a week during the school semester and a full-time over the summer and vacations. I was working at the time at Environmental Analytical Testing Laboratory, Analytical Resources Incorporated. And it was a great experience because that got me started on a real analytical chemistry career. So we were doing a regulated environmental analytical testing for a wide variety of facilities and organizations across the Washington state and also the Pacific Northwest. At ARI, I had an opportunity to really see what a career field in analytical chemistry looked like. And that was an emphasis of my focus at the University of Washington. But I also had an opportunity to do some undergraduate research in the lab of Isiao Packer. And Professor Packer got me started on working with fullerenes, C60 and C70. I had a great time working with Mike Corey and Greg Spiritus in Dr. Packer's lab, where we separated C60 and C70 from a raw material batch using liquid chromatography. And then we also did several molecular modeling studies to understand the highest molecular orbital and the lowest molecular orbitals of the C60 molecules, which look like a cage if you haven't already seen them. I had an opportunity to present that undergraduate research And part of the benefit of that was having some nice experience and a professional poster on my resume when I sent it out for jobs just before my graduation, which that was the end of 1993, I think. Fortunately for me, I had an opportunity that my resume attracted the interest of the metabolism team at Pfizer Central Research in Groton, Connecticut. And they were interested in me, not just because I had an analytical chemistry background, but also I had had an opportunity to work with animals in a couple of jobs previously, one working on a farm for thoroughbred racehorses, and another job working for a veterinarian. And in late 1993, I had an opportunity to interview. They flew me out to Groton, Connecticut, which is quite a distance away, in a winter snowstorm. And I must have impressed them somehow, because they certainly gave me an excellent opportunity to start work there in January. So we moved between Christmas and New Year, 1993 to 1994. And I started in early January 1994 in the drug metabolism group doing pharmaceutical research. Now, drug metabolism, for those of you who don't know, is the study of how the body interacts with a new chemical entity that's proposed as a drug candidate. So there are two ways to look at a medicine. The first way to look at it is what effect does it have in terms of treating the disease? And the second way to look at it is what effect does it have in being cleared from the body when given as a medicine? And that's the science of drug metabolism. So I spent the first couple of years working on a project team doing traditional pharmaceutical discovery. And we were working together with biologists and chemists, metabolism scientists, some toxicologists, some pharmacologists, some folks who were expert in chemical synthesis in scaling up, making a new compound, and some other folks who were experts in other parts of the biological systems that we were using as models. And we were testing molecules that were similar to our candidate molecule to find what you typically term of as the lead candidate, and then its backup, a compound that you would advance in tandem as much as possible, and then substitute if for some reason that first lead compound failed for one reason or another. We got as far as the early toxicology studies before, unfortunately, a toxicological event occurred. And so we did a series of studies thereafter to investigate that event and to see if the backup candidate did not have the problem. But unfortunately for the whole program, it turned out that the problem was a little too challenging to address immediately. And from a market perspective, the particular program was canceled. So after an appropriate period of mourning, which in fact included a wake for the project team, I started work with another group doing higher throughput screening and looking at in vitro models of drug metabolism in both animals and humans. Those models include hepatocytes, which are individual cells that occur in the liver. And then it also included tissue slices of liver and kidney. And we would use these models to evaluate, to try to predict how a compound would be cleared by the liver or the kidneys in a human system without having any actual human in vivo data. So we'd take a look at the animal in vivo data and we'd say, here's what the animal in vitro data, the slices or the hepatocytes would predict. And then we'd look at the in vitro human tissue data, and then we'd say, well, what does that mean then? How would we think about scaling that up to being in the actual clinical situation? So I spent a couple of years working on that model with a team. And it was right about that time that most of the major pharmaceutical companies got very interested in understanding the chemical and the physical chemical properties of all the compounds that they had in their chemical libraries. And for the big pharma, these were quite a few. At Pfizer at the time, there were over 600,000 different compounds, and more were being added every week. So I joined a team to start working on setting up higher throughput screens using these in vitro hepatocytes or slice models. And we eventually settled on using both hepatocytes, the actual cells, and a subcellular fraction called microsomes, which is kind of a fancy term for when you grind up the microsomes and then you filter them in a way that gives you just the part that has the metabolizing enzymes. So there would be the metabolizing enzymes separately, and then the cells where the drug would have to cross the cell barrier to reach the metabolizing enzyme. So first I worked a little bit with the actual models, the in vitro models, but then I quickly graduated to using my analytical chemistry expertise with a team that was working on analyzing the samples from these models using HPLC with mass spectrometry analysis. And HPLC is an acronym that means high performance liquid chromatography, which is a way to separate compounds that are in a mixture so that you can detect the individual compounds. And the mass spectrometry was just the detection by the mass of the actual molecule of interest, the candidate compound. So we had to come up with a way to introduce samples, 10 or 15 at a time, into a mass spectrometer, make an individual measurement that reflected each one of those, the concentration of the compound in each one of those samples, and then turn that data around and calculate what we call in vitro clearance, which is really the loss of the compound over time. And our biology colleagues were creating the actual samples. They were doing the incubations, and they would send them to us in 96 well plates. And we took those plates and put them on instruments that we built or assembled rather out of 2 or 3 other instruments, got them all to communicate together so that we can make the analyses, produce the data, and then send them on to our database colleagues who would set them up in the database where it would automatically do the calculation. So after about 8 months of work, we reached a point where the whole team was analyzing about 1,000 compounds through these screens a week, and then uploading all of the data along with a bunch of other data about the physical chemical properties of those compounds into the database. So Pfizer project teams could query it and say, is there a compound in here that we could potentially use for a medicine that would make a good medicine, right? Does it have the right physical chemical properties? Does it have the right biology that we would need? All of those pieces. So that was pretty hot research at the time and quite a lot of fun, but I recognized that there was that I wanted to do something a little bit different. And more importantly, I wanted to move back from the East Coast to the West Coast, back home, if you like to put it, where family was. And so a few trips to the coast and talking to some other folks eventually led us to an interview at contract research organization, MDS Pharma Services. And that organization did a lot of things, but the unit that I interviewed with was expert in pharmacology discovery screening. And what that meant was they were looking for off-target pharmacology effects for a new chemical medicine. So when you are picking a new medicine to advance as a candidate, you're looking for your primary pharmacology event, and you want to try to eliminate all the other events as much as possible, because they're the ones that tend to cause side effects or potentially toxicology events that could kill your candidate. So pharmacology screening across a broad range of different tests provided that information. And the company also wanted to provide some services for this in vitro metabolism screening that I had been doing and was expert in. So in 2001, I started work at MDS Pharma Services in Bothell, Washington, just north of Seattle. And it was a great and exciting time. Certainly that first year was pretty exciting since we experienced the Nisqually earthquake in February, and then unfortunately 9/11 in that September. But neither of those things proved predictive of any disaster at MDS. In fact, I went on to spend another 6 years there working in the drug metabolism and pharmacology range, and eventually being promoted to a technical director of pharmacology and metabolism, one of the more senior roles. And in that role, I had an opportunity to work with our sales colleagues to talk to new clients and understand their needs, and then design studies to help answer the questions they were asking about their compounds with respect to the pharmacology, both on target and off target, as well as the drug metabolism and their drugability properties overall, right? Would does this compound make a really good medicine? Excuse me. So, but closing around 2010, we recognized that the demand for elder care responsibilities was going to mean that we needed to move back home to Spokane. So started looking for opportunities, and very fortunate to find one at our own homegrown Jubilant Hollister steer. JHS, as we affectionately refer to her, is a sterile drug products manufacturer, and that means that they fill vials, sterile vials, with liquid or lyophilized, freeze-dried drug products, and those drug products are then sold. They're then labeled and made available for prescription medicines. My analytical chemistry expertise attracted their attention and eventually led to a job offer in the quality laboratory here at JHS in Spokane, where I spent a number of years as a senior scientist and supervisor of a team of quality control scientists. And that was a great experience. We did lots of very interesting work at Jubilant, and much of the work that they do is to create medicines that many may be familiar with, in the categories of imaging agents that might be used for sonography or radiography, and also vaccines. And very proud to say that I was part of the team that was working on vaccines for our recent pandemic, as well as treatments for the pandemic. But after about 2015 or so, I realized that I needed to grow a little bit more, that I wanted some different career choices. And part of those career choices meant making my own choices about my career, as opposed to being in corporate America. And that led to me forming my own professional services firm in 2017. And in that work, I use my expertise to help executives, entrepreneurs, investors, and leaders get their medical products, both medicines and medical devices, from the bench to the bedside. And how do I do this? I work with them on individual projects. So is much the same way that I worked with people at a contract research organization, MDS Pharma Services. I have an opportunity to meet people who have problems. They've reached a point in their development of their medicine or their medical device where they face a challenge. And quite often I've got some experience facing a very similar challenge throughout my career. So I can use that experience, but I can also use my very deep network to find service providers, to introduce them to other experts who may have more specific knowledge than I do, and to generally help them work through the challenges that everyone faces when they're developing a medical product. This is a long game after all. It's not like software, where quite often the timelines are very quick. 6 months, you've got a product, and in another 6 months, you've got your almost final product and you're ramping up your sales very quickly. No, in fact, for medicines, for new medicines, it typically takes 13 years from the filing of the patent to get to launch with a successful compound. And the attrition is pretty deep along the way. You can start with about 40,000 compounds in discovery and end up with about 200 in preclinical. And from there, you drop into 13 that actually advance into clinical studies. And out of those 13, you're lucky if you get one. So, and it's not always 100% that you will get one through that 13 years. Similarly, in medical devices, there's a somewhat shorter lifespan. So it typically takes about 6 to 7 years from the filing of the patent to get to a product launch. And the attrition rate's a little bit better in medical products, but it's still definitely there. It tends to occur less on the actual ability of the product to do the job, more on whether or not the market will be paying for the new product, or whether it's actually more cost effective or better than the current standard of care. So that's the work that I do in my professional services firm. As an entrepreneur, which is what I am, I do own my own business, and I've had an opportunity to interact with other entrepreneurs in the Spokane area, and that's a very growing community. I had the opportunity and the pleasure of meeting Professor Glenn Prestwich of Washington State University. Professor Prestwich was at the time the President's Distinguished Professor for Washington State University Spokane here in the Health Sciences campus. And he was charged with helping Chancellor DeWald improve the entrepreneurial aspects of the WSU community and overall here in Spokane. Professor Prestwich helped introduce me to the College of Pharmacy and Pharmaceutical Sciences and facilitated my entry there as an adjunct faculty member. And that's been very enjoyable since I get to work as a principal investigator on talent development objectives. And that's a passion project for me because I recognize as a hiring manager that you don't always get the opportunity to be introduced to the candidates that you ideally want to see. And as you all recognize too, from where you stand, candidates who are coming through and taking a baccalaureate degree may not have all of the information that they need to know for their first corporate role, for their first career role. And so working together with some colleagues in the Project Lead the Way organization, as well as Washington State University, we are working to build a pipeline for students out of high school towards either their post-secondary education or their initial post-secondary career, facilitating the biotechnician assistant credential and exam as an industry recognized credential that helps people understand what it's like to work in the biopharmaceutical manufacturing industry and in other life sciences industries that are very similar. I'm also part of a group that works to develop entrepreneurial resources. We call ourselves the Flexible Infrastructure for Resilient Entrepreneurship because we enjoy our acronyms. That one is FIRE. And I'm the leader of the Workforce Innovation Network Development, or WIND team, under the FIRE grant. I also have opportunities to work with colleagues across Washington State University through our local incubator, SP3 Northwest. And it's SP3 because carbon is SP3, but it is a life sciences incubator that was founded here in Spokane and operates out of the WSU Spokane campus, but has satellite facilities at all of the campuses across, all of the WSU campuses across Washington State. So pretty excited by the entrepreneurial opportunities that are available for people with an idea for a medicine or a medical product or something that will generally help in the life sciences space. And I've also had the pleasure lately of working with the team, including Professor Prestwich, to build an innovation cluster here in Spokane, centered here in Spokane, but we think of ourselves as being a more regional play, knowing that contract services are very important for new and virtual entrepreneurial companies in medical products, both medical devices as well as pharmaceuticals, and recognizing that the Pacific Northwest is a little short of some of those resources, and that the resources that are out there available nationally and internationally there may have been a little too much consolidation for them to be accessible for smaller companies. Nevertheless, smaller companies represent an ever increasing fraction of the discovery work that's being done in pharmaceuticals and medical devices. So there's a real gap for these companies when they go to seek contract services for the development of their products. If they're, for example, if they don't have $1 million to spend on a study, the CRO may not even return their calls. Yet they still need this. They still need a study in order to get the data that they need to advance their product. So our innovation cluster, which is titled Evergreen Bioscience Innovation, will work to build those contract research resources, starting here in Spokane and expanding outward in ever widening circles. We're working with colleagues in Idaho and Montana, as well as British Columbia and Oregon, to fulfill those needs. And a couple of our core members, of course, are right here in Spokane, Jubilant Hollister Steer, as well as Selkirk Pharma. So again, our vision would be that here in the Mountain Northwest, in all of these states, we have the infrastructure that could take an idea from its very germination all the way through to full development within our own space, without having to go back east to the classic New York, New Jersey, and Pennsylvania tri-state area for development, or even Research Triangle Park, or perhaps south to San Francisco or San Diego. We think that our region is really well placed for this. We've got a great quality of life. We've got a good cost structure for new companies to move in, and we're certainly attracting them as well. So we're pretty excited by all this opportunity, and we're working as well with Life Science Washington to help make this dream a reality. So that's kind of the space in which I've been operating and where I'm at today. What's going to happen tomorrow? I don't know. It's going to be fun to find out.